All right, well, hi everyone. I'm an NIH predoctoral fellow in Tony Wilson's lab at the University of Nebraska Medical Center in the US. And one of the main focuses of our lab is to use physiologically informed models of aging to try to understand what we think are important questions in cognitive neuroscience. So I'm going to go over one of those studies with you today. But first, I think I need to address sort of an important topic, which is the question of what are we trying to actually study when we study aging, when we operationalize age. At first, that seems intuitive. Age is the amount of time someone has been on the earth, right? But when you think about it, that's not typically what we're actually trying to get at usually. That's sort of secondary. Really what we're trying to measure is the effects of accumulated physiological stressors on the human body or the human mind. Taking that into consideration, chronological aging metrics, be them in days, months, years, are actually pretty poor predictors of our primary outcome measures in aging, which are our morbidity and mortality. So more anecdotally, I can ask you all, what does the age of 70 look like, right? And I'm sure some of you will think it looks like this, others will think it looks like this, and others will think it looks more like this. To address this kind of disparity between our physiological measures and our functional outcomes of some of the things that typically are shown to decline with aging, scientists have really turned to these biologically based or physiologically based metrics of aging to help inform their models. And one of the more popular of these recently is DNA methylation. So I'm sure you all came here for a refresher on intro genetics, so you're welcome for this. But basically, DNA methylation, for anyone who's not up to date, is an epigenetic regulation of the genome. So I'm sure you all know that every cell in your body has the genetic information for every other cell in your body, and basically genetic regulation is what differentiates cell types. So epigenetic regulation has been found to vary not only by cell type across tissues, but also as a function of age and as a function of a number of important physiological or environmental factors, particularly things like socioeconomic status, HIV infection, and others. So recently, scientists have been able to sort of leverage this pattern of differential epigenetic modulation to build really accurate and informative models of aging that we can use in our models. So as you'd expect, these models typically co-vary pretty strongly with chronological age. This is actually data from the experiment that I'll be showing in a minute. But plotted in blue here, what you see are the sort of the relationship between chronological age and our methylation metric of aging in our sample. And as you can see, the fit line there represents a pretty strong relationship of 0.94. But what we're actually interested in here is the divergence from this fit line. So you can imagine someone who falls above the fit line, like this person, would be biologically older than what we would expect from their chronological age, while someone around the same chronological age falling below the fit line would be biologically younger than what we would expect. And we refer to this as age acceleration or deceleration, typically. Now, this is important because this really tells us what we're getting from these biological aging metrics above and beyond the effects of chronological age. You can imagine an IRB doesn't like us poking people with needles if we don't have to, and we really need to sort of show that these are giving us something above and beyond chronological aging, because it's obviously much easier to ask people their birthday than to pull whole blood samples and sequence their epigenome. So we wanted to look at the relationship between this accelerated aging metric and some type of cognition. So age, as many of you know, tends to exhibit subtle declines in a number of cognitive functions. So among these are memory, motor function, and selective attention. We chose selective attention here for our first study just because it's pretty well established to decline subtly with age, and it's very amenable to neuroimaging research in general. So I'll blow through this pretty quickly, but we had 68 participants in this study between 22 and 72 years of age, pretty evenly distributed along that spectrum. We used magnetoencephalography, or MEG, for our functional neuroimaging. This is because it allows us to not only look at the spatial distribution of our neuroresponses, but also the spectral and temporal dynamics, which we were interested in here. And we had participants perform 200 trials of an arrow-based flanker task, which many of you will be sort of already familiar with, but I'll go through it in a second for those who are not. We had structural T1 data for co-registration of all of our functional data for every person, and we importantly used a really established and well-researched modeling pipeline for our DNA methylation age estimation. So we collaborated with Trey Eideker's lab at California, San Diego for this. He's pretty established in the field, and they've been working for the last over a decade, I believe, to develop these really sort of complicated algorithms that I'm not sure I fully understand, but I've got a good grasp on for modeling age biologically from whole methylome data. So this is the task that we had participants perform. It's just a simple Erickson flanker task, an arrow-based Erickson flanker task. Participants fixated centrally for our baseline period, and then were presented with one of these 4 rows of arrows that you see here. Importantly, they were only responding to the direction of the central arrow in this array, and the 4 surrounding arrows or flanking arrows are completely task irrelevant, or at least irrelevant to their response. So sort of the typical finding here is that despite those flanking arrows being not relevant to their response, participants are much slower when responding to the incongruent compared to the congruent trials. Typically what you do with this data is you actually subtract whatever metric you're looking at, be it reaction time or neural data, you subtract the congruent information from the incongruent information, and that gives you the absolute effect of that distraction, of those distracting arrows, again, on behavior or on neural metrics. So first, we wanted to make sure that our aging metrics showed sort of the typical relationship you see with this selective attention function. So on the left, you have chronological aging on the x-axis, and on the right, you have the biological aging on the x. And on the Y here for both of them, you have the behavioral flanker effect, again, just indexing sort of that distractibility effect. And so on the left, you can see we saw sort of the typical pattern, the typical relationship between chronological age and selective attention, where older adults were more distracted by the flanking stimuli, nothing new there. But when you look on the right, we actually interestingly found that biological age showed the same effect and actually also predicted the selective attention function above and beyond the effects of chronological age. So already a good indication here that we are getting something above and beyond chronological aging here. We didn't poke a lot of people with needles for no reason. This part I'm not going to cover in as much detail for time constraints, but basically we looked at all of our activity, our neural activity in the time frequency domain, which allows us to look at these different time frequency responses that are known to have different functional significances. And we saw 3 really well-defined and well-studied responses here, an early theta band response, a later alpha band response, and sort of a temporally overlapping gamma band response. We took these time frequency definitions and did what's called beamforming to image them to the level of the cortical surface. And then again, what we did is we took those whole brain voxel-wise maps and subtracted them between the congruent and incongruent condition to give us a measure of that distractibility or that selective attention function per time frequency response. We then regressed those maps on that age acceleration metric that I was talking about. Another way to say that is that we looked at the effects of biological aging on selective attention function in the brain above and beyond the effects of chronological aging. So interestingly, the only significant effects after multiple comparisons were in the gamma band, which is kind of cool but also difficult to explain in a lot of ways because a lot of cognitive interference tasks have really only been looked at in EEG, and EEG has trouble sort of looking at some of the really high frequency activity that we defined in this task. So the map you see here is actually the whole brain regression that I talked about. And as you can see, there's sort of an anterior cingulate peak that was very robust and survived very stringent multiple comparisons correction. The relationship from that you can see on the bottom left there, where adults who were biologically older than you would expect them to be from their chronological age had more of an effect in the anterior cingulate cortex and the gamma band on those interfering trials. Interestingly, you can see on the bottom right, activity in this region also correlated with the behavioral effect. Not a very strong correlation, I know, but it was significant in our data. And activity in this region actually mediated the relationship that we saw on an earlier slide, which I can go back to, between the biological aging metric that we see on the bottom right here and the behavioral effect. So really tying together nicely sort of this behaviorally relevant link. So I kind of flew through that, but to conclude, our favorite measures of aging based on chronological count variables are not aging very well. And I think the inclusion of biologically and physiologically relevant metrics into our aging models to try to predict sort of age-related declines can hopefully account for some of the variance that we're seeing. Towards this goal, we found a robust and behaviorally relevant link between biological aging and the neural dynamics that support selective attention. And this is important because DNA methylation, again, we do have to do a whole blood draw here, but it is much less invasive than some other biological aging metrics that are currently used. And it does seem to supply some novel information above and beyond the effects of chronological aging that will hopefully assist in our aging models and our research. So to conclude, I need to acknowledge my great lab and my PI for this, as well as our collaborators at UNMC, specifically Howard Fox, who did a lot of the methylation analysis, and our collaborators at UCSD in Trey Idicker's lab, our funding sources, and also a shameless plug, we're hiring and always looking for motivated research assistants, grad students, and postdocs in our lab. So if you're interested, just speak to me after the session.